electron acceptors such as oxygen to drive the bulk production of adenosine triphosphate or ATP, or the energy currencies which contains energy. Cellular respiration may be described as a set of metabolic reactions and processes that takes place in the cells of organisms to convert chemical energy from nutrients into ATP and then release the waste products also. The reactions involved in the respiration are catabolic reactions, that is, which break up large molecules into smaller ones and finally results in the production of energy. In the diagram shown here, you can see, or the flow chart shown here, the glycolysis in cytoplasm is divided into energy investment stage of the first 3 steps, and the last 3 steps are the energy harvesting stages. In the energy investment stage, you can see when the glucose, when it is getting converted to glucose 6-phosphate, one ATP is being utilized and converted to ADP. And when glucose 6-phosphate is converted to fructose 6-phosphate, and then finally the fructose 6-phosphate is converted to glyceraldehyde 3-phosphate, that is 2 molecules of glyceraldehyde 3-phosphate will be formed, and this will again require an energy conversion, that is ATP to ADP. Then this glyceraldehyde 3-phosphate, the 2 molecules, when they get converted to 1,3-bisphosphoglycerate, an NAD is getting converted to NADH. And 1,3-bisphosphoglycerate, when it gets converted to 3-phosphoglycerate, and ADP is getting converted to ADP, ATP, that is the energy harvesting stage has started. And this 3-phosphoglycerate, when it gets converted to phosphoenol pyruvate by the removal of a molecule of water, and ADP is removal of water, and 3-phosphoenol pyruvate, when it gets converted to pyruvate, again one ADP is getting converted to an ATP molecule. So that as 2 molecules of 1,3-bisphosphoglycerate is involved, 2 ADP is getting converted, total of 4 ATP molecule production is happening. But as we have already utilized 2 ATP molecules during the energy investment stage, so a net gain is only 2 ATP molecules. So we have 2 NADH molecules and 2 ATP molecules from the glycolysis, which will enter into the electron transport chain for the further ATP production. Whereas from the citric acid cycle, you can see there is like 2 ATP molecules production and 3 NADH and 1 FADH that is produced, which also will further enter into the electron transport chain and results in the production of ATP. So respiration is one of the key ways a cell releases chemical energy to fuel cellular activity. Nutrients that are commonly used by animal and plant cells in respiration include sugar, amino acids and fatty acids, and the most common oxidizing agent is molecular oxygen. The reactions with a negative delta G release means that they are processed without an energy input or they are spontaneous, and the negative delta G indicates that reaction is exothermic or exergonic and can occur spontaneously. In contrast, reactions with the positive delta G need an input of energy in order to take place, they are non-spontaneous. So it's been told that 38 ATP molecules can be made per oxidized glucose molecule during cellular respiration, that is 2 from glycolysis, 2 from Krebs cycle, and about 34 from the electron transport chain when NADH and FADH from glycolysis and citric acid cycle enters into the electron transport system. However, this maximum yield is never quite reached because of losses due to leaky membranes as well as the cost of moving pyruvate and ADP into the mitochondrial matrix. And current estimates range around 29 to 30 ATP molecules are the actual ones that are produced per glucose molecule. So in this table you can see glycolysis in the preparatory phase we use 2 ATP molecules. That is, the source of ATP here is phosphorylation of glucose and fructose 6-phosphate, which uses 2 ATP from the cytoplasm, that is 2 ATP, that is -2 it is shown over there. Then glycolysis payoff phase, or where the energy yielding phase is 4 ATP molecules are produced as a result of substrate level phosphorylation, and 2 NADH molecules are produced and from which 3 or 5 ATP molecules can be produced by oxidative phosphorylation. Then oxidative decarboxylation of pyruvate, where 2 NADH molecules are produced. There again 5 ATP molecules yield can happen because of oxidative phosphorylation, and in Krebs cycle 2 ATP molecules are produced by substrate level phosphorylation, and 6 NADH molecules which are produced can give rise to 15 ATP molecules by oxidative phosphorylation, and 2 FADH2 molecules which are produced during Krebs cycle can give rise to 3 ATP molecules by oxidative phosphorylation. So a total yield of 30 or 32 ATP molecules from the complete oxidation of one glucose molecule to carbon dioxide and oxidation of all these reduced coenzymes can happen. So it's not, actually speaking, it is not 38, but 30 or 32 ATP molecules are actually produced. Thank you.